Good morning, everyone. Thank you, John, for inviting me, and thank you, the organising committee. So in this talk, I'll be discussing how the Alzheimer's-linked amyloid beta or A-beta proteins can provide new insights into the mechanisms underpinning retinal degeneration. You might be interested to know that carotenoids have been shown to dampen down some of the cytotoxic effects of A-beta. And there's also evidence to show that carotenoids can prevent the aggregation of A-beta as well. Those who are not familiar with A-beta, they are largely found initially as soluble monomers. These are alpha helical or unordered structure. They self-associate to form dimers and trimers, and it's the oligomeric form that is most closely associated with Alzheimer's pathology. These aggregate to larger structures to form protofibrils, which are rich in beta sheets, and deposit as mature fibrils in brains of Alzheimer's patients. And we can study this in vitro. You can see the aggregation of A-beta 1 to 42, which is the most aggregate prone of these molecules. You can see the self-association of these starting from monomers into ever increasingly larger structures. Now, over the past 20 years or so, a large body of work from a number of labs have shown the involvement of A-beta in the aging and diseased retina. And this is using a variety of antibodies that detects different forms of A-beta as well as probes. And what I've done is I've picked out a few of these papers. Here, 2004, showing the presence of amyloid vesicles in drusen and how these the different forms of A-beta are found in these drusenoid structures. Evidence that these amyloid beta deposits in drusen associate with complement proteins that are also associated with retinal degeneration. And a body of literature showing that moderate to high amount of drusen, as well as drusen in patients from AMDIs, have large amounts of amyloid beta in them. More recently, Robin Guymer's group showed how plasma levels of A-beta can be used to predict late-stage forms of AMD, particularly the wet or the CNV form. And here, this is work from Professor Glenn Jeffrey's work showing the involvement of A-beta and how it accumulates in the mouse. So the pattern of A-beta accumulation can be recapitulated in mouse models. And here's some more evidence of how the pro-inflammatory, pro-angiogenic effects of A-beta can be studied using zebrafish, for instance. A few more papers showing how exposure of RPE to A-beta can alter the structure. And here, association of complement proteins, again with A-beta. And some studies from Kao et al. And Matsubara's group showing how exposure of A-beta, RPE cells to A-beta, changes their cytokine profile to bring about a more pro-inflammatory phenotype. The deposition of cholesterol and intake of high-fat diets has been shown to promote the deposition of A-beta as well. And here, a range of examples here, this one from Catherine Bowes-Rickman's group, showing how the use of anti-A-beta immunotherapy can rescue mouse models and rescue their vision. And this one is another example where high-fat diets has been shown to promote the deposition of A-beta alongside ApoE. If you look at this picture from the side of the brain, so I've just picked out one paper, but there are lots of mouse models of Alzheimer's disease, and they have inner retinal defects. And here, an example where amyloid plaques have been shown in retinas from Alzheimer's patients. our own work showing how retinal neurons take in A-beta and how the dynamics behind it and how it can alter the structure. And here we've looked at the evidence of the vast amount of literature that's come out showing visual defects in patients with dementia. And this is something that's not very well reported and even less poorly understood. So we still have some unanswered questions. For instance, we don't understand how A-beta affects living eyes, living tissues, particularly the outer retina, because most of these studies have been done in fixed tissues. So we want to understand the dynamic effects associated with A-beta-driven pathology. And what are the effects at single cell level? We're particularly interested in the RPE and the choroid, given their importance in early disease. So to address these questions, we've developed a mouse model that recapitulates the A-beta burden in living aged disease retinas, and also we've turned to cell models to study A-beta effects at single cell resolution. In preparing A-beta, we used pico-nanomolar concentrations and only up to 1 micromolar, which has been reported in ocular fluids. We used human oligomeric A-beta, and this is the conformation and species most closely associated with Alzheimer's pathology. And we prepared A-beta using a very well-established protocol, and we've tested this using negative stain EM as well as immunogold labeling. And also tested these for endotoxicity. And you can see 1.5 hours after preparing A-beta, we find the formation of oligomers, which corresponds to the size of reported oligomers that's been reported in the literature. So all these experiments were done using human oligomeric A-beta prepared 1.5 hours after preparation. So yesterday you heard some great talks about how mouse models can be exploited. So here's another example of that. So here we've used C57 black 6 mice, and we've given these mice a single injection of subretinal A-beta. This is quite a nice model because it offers some advantages over some of the Alzheimer's transgenic mice, because we can control the type of A-beta, the species and the conformation that goes in, regulate the location and the extent and the duration of treatment. So we did baseline ERGs and OCTs on these mice. 8 days later, we carried out subretinal injections, or one injection, or either vehicle or A-beta. We immediately tested this using fundoscopy. And here, 8 days later, ERGs, OCTs, and fundoscopy. And a cohort of these mice were culled, taken to histology. Another group remained 8 days later, ERGs, OCTs, and again they were culled. So what did we find? We first looked at color fundus photography, and you can see here the presence of successful injection. The retina becomes detached, and then 24 hours later it reattaches. But if you look at a week later, you can see normal pathology, the normal retina. But in the A-beta injected eye, we see what appears to be some superficial pathology. We don't know what that is. We also did full-field ERGs on these, and we were a bit surprised to see that there was no effect at all. It was reassuring in some ways because we hadn't destroyed the retina in any way by doing the subretinal injections. And we sort of discovered why there were no effects, because we found that so these are OCT images, and I pulled out some pictures for that to illustrate the point. The damage that was done by A-beta appeared to be very localized. So a full-field ERG is unlikely to pick this up. And I will highlight some features. You can see, particularly in week 1, the accumulation of subretinal fluid and some hyper-reflective material. We see some RP pathology here. By week 2, this subretinal fluid seemed to have resolved largely, and we find the appearance of hyperreflective spaces appearing, particularly in the choroid. When we looked at the histology, we found disrupted and atrophic RPE, disorganized inner segments, and the absence of outer segments on occasion. And we found these areas where we think would have corresponded to areas where fluid may have accumulated subretinally and in the choroid, as you just saw in the OCT images. Looking at immunofluorescence, we see again disrupted outer segments and really the pathology at the RPE photoreceptor interface. We did some morphometric analysis, and we pull out the sort of data showing RPE pathology and certainly a trend showing changes in outer segments. So there's definitely something going on with the mouse and in the choroid. So we wanted to look at this further, and we turned to culture models. And here is an RPE culture model using the transfer system, which we have characterized to a great extent, and lots of other labs use this as well. And previous findings showed that exposure of RPE cells to A beta appear to mess around with the effects of that the amount of VEGF and PEDF secreted by RPE. We found when we used physiological concentrations, in contrast to some of the previous doses that's been used, which wasn't really physiological, we found that VEGF and PEDF levels remained unchanged. We do report for the first time that A beta is secreted directionally by RPE cells, and that there was a predominantly basolateral secretion towards the choroid, 1.8-fold over levels secreted into the apical compartment. And this links quite nicely with the choroidal pathology that we saw in the mouse model. So we wanted to look at the choroidal effects a bit more. We turned to a choroidal endothelial cell model, and here is a scratch assay, and you can see how vehicle versus A beta, and we wanted to find out if there was a change in the way the scratch gets recovered by cells. And what we find is exposure of choroidal cells to A beta resulted in an altered behavior of endothelial cells. And this is either a cell proliferation or migration. Without doing further experiments, we can't tease those two apart. And this, again, ties nicely because the effects were observed at delayed time points, 24 and 48 hours, and ties nicely with the delayed choroidal effects we saw in the mouse model. Looking at the RPE cells, we asked what effects does A beta have in the RPE barrier. So we treated RPE cells with A beta, and we looked at the morphology of the RPE monolayer. We saw what appeared to be lesions in the RPE, and I was struck by this because Professor He-Ping Hsu's lab has shown the formation of these RPE lesions in C57 black 6 mice that are 18 months old. So knowing that the barrier is disrupted, we wanted to see whether that has any consequences, and we did a FITC-dextran assay to see how A beta, whether the permeability of A beta has been changed in any way, and we find that at 48 hours, the change in morphology has interfered with the RPE barrier function. And these findings are consistent with pro-angiogenic and reduced barrier integrity that's been reported in Alzheimer's patients. We asked, is A beta internalized by RPE cells? And if that is the case, what has happened to it? And this is a system that we have characterized very well, where we feed RPE cells with photoreceptor outer segments, POS, tagged to FITC, and we've shown how they're internalized via MERTK and alpha beta-5 integrin, and how these cargoes are trafficked through early endosomes, late endosomes, lysosomes, and ending up in late-stage autophagy bodies. And these data are consistent with reports shown by Silver Feneman's group. So we asked, well, what's happening to the A beta? And we show for the first time that A beta, RP lysosomes accumulate A beta, here using lysosensor. And as many as 40% of RPE lysosomes contained A beta. And we also noticed that these A beta containing vesicles had an enlarged and swollen phenotype. We asked then, how does the RPE cell respond to lysosomal A beta? So here's a new cargo that's arrived in lysosomes. How does the cell respond? So here's the assay. We pulse A beta. This is washed away. and then we image at these time points the activity of cathepsin B. This is lysosomal cathepsin B, and we wanted to find out how it responds. So here you can see magic red labeling lysosomal cathepsin B, and here A beta tagged to FTSE. And what you find here is that very early on, when cargoes arrive, the lysosome responds by increasing cathepsin B by as much as 153% over baseline. By 3 hours, this has diminished to 112%, and then it tapers off. Then 24, 48 hours, there's no difference because it's returned to baseline levels. Well, has it done the job? What is the effect of lysosomal B cathepsin upregulation on the A beta cargo? Here, we looked at the A beta signal, and you can see at 0.5 hours after treatment how A beta is accumulating in RPE lysosomes. And then 3 hours later, there's another 7.5-fold increase. And here, 24 hours later, we can see a further 17% increase. So in our assay, this is the maximal point of A beta aggregation in RPE lysosomes. But you know at this point, the early time points is when cathepsin B upregulates, and by 48 hours, the level of cathepsin has come down. So in effect, by 48 hours, from a maximal time point, cathepsin B and other lysosomal enzymes that are there have only managed to reduce A beta by roughly 16%. And in fact, 84% of A beta in RP lysosomes remains sequestered. And this is the first time this has been shown. And what is the nature of this upregulation? What is the nature of cathepsin B upregulation in response to lysosomal A beta? We did some qPCR analysis, and it showed that actually there was no change in the mRNA of cathepsin B. This is interesting because the enzymatic half-life of cathepsin B is only 14 hours. And if there was a change at the level of gene regulation, we would have seen some changes, and there's no change at all. So the cellular response was not at the level of gene expression. I apologize, this seemed to have just merged with that. Finally, we asked, what are the functional consequences of A beta in RPE lysosomes? Does it actually have an effect on A beta? So one of the biggest jobs of the RPE is to internalize photoreceptor segments on a daily basis. So the proteolytic load on the lysosomes of the RPE cells is considerable, and one of the highest in the body. So the assay, we pulsed A beta and then washed away. 24 hours later, these RPE cells were given a feed of photoreceptor segments. And then at time points that are consistent with work that we've done, and certainly data from Sylvia Finneman's group, we looked at how POS trafficking was affected. So what we find, we used the Costes method. This is a non-biased quantification of co-localization, and we looked at POS labeled with FTSE, and also lysosomes labeled with LAMP1. And what we find is that certainly 4 hours after POS, there's no difference in the number of lysosomes that are participating in POS trafficking. But certainly by 8 and certainly 20 hours, there's a 20% reduction in the number of lysosomes that are participating in POS trafficking. And this is the effect seems to be pronounced at the time points that's been shown where POS cargoes are degraded by RPE cells. So let's summarize. Using the mouse model, we looked at OCT and we showed how A beta effects in the living mouse eye change. We saw subretinal hyperreflective material, fluid accumulation, changes to the RPE, and this is consistent with a CNV phenotype that's been reported in these papers. We saw these hyperreflective spaces in the choroid. This is 2 weeks after A beta exposure, and this was correlated with diminished subretinal fluid. So this is really evolving pathology that you really see for the first time. And certainly there's the role of the choroid that's been shown here. Looking at histology, we can see disrupted atrophic RPE, obliterated outer segments, and areas where fluid may have accumulated. Combining mouse models with the cell models, we see that these pro-angiogenic A-beta effects in the choroid appear to be directly mediated rather than through VEGF, which was considered to be the case by some scientists before. And the evidence for that, you see predominant A-beta secretion via the basolateral RP surface. We see disrupted barriers. And this is something that's been reported by these studies in mouse models as well as cultures. And you saw evidence how choroidal endothelial cells and how their behavior can be changed by RPE cells, particularly delayed time points. And this is consistent with the data from delayed effects in the living mouse eye. And evidence that A-beta is co-localized to RPE lysosomes, as many as 40% of vesicles. Now, this is interesting because as far back as 2004, Ralph Nixon's group speculated that the aggregation of high molecular weight molecules in lysosomes of neurons could be a major factor in triggering Alzheimer's disease. And since then, there's been some very nice biochemical studies showing how A-beta is inserted into lysosomes of neurons. And certainly, lysosomal pathology is one of the early hallmarks of Alzheimer's disease if you look at postmortem brains. We see here lysosomal A-beta accumulation associated with upregulation of vesicular cathepsin B. So this is a cysteine protease. This is quite important. Most people think about cathepsin D, but actually cathepsin B is also important because lipid peroxidation products in the aging eye, the H&E and MDA, has been shown to have profound effects on cathepsin B activity, reducing their activity by as much as 90%. Mike Bolton's group showed how lysosomal enzymes, the activity are highest in RP from the macular region compared to RP from the peripheral retina. Again, perhaps indicating that the effects may be particularly pronounced in the macular region. And we saw that lack of change in cathepsin B mRNA suggests the enzymatic upregulation was mediated perhaps at the level of proteins. But you saw that despite this, A-beta remains sequestered within lysosomes after cathepsin B had returned to baseline levels. And this also showing that enzymatic activity degraded only 16% of A-beta. So this suggests impaired cargo clearance by cathepsin B or other lysosomal enzymes. There are as many as 60. We didn't measure them, but we measured the cargo, and we see that A-beta still persists. So this suggests an altogether new mechanism where A-beta can accumulate in RPE lysosomes over the life course. And finally, the consequences of lysosomal aggregation in RPE. We see diminished numbers of lysosomes, as many as 20% less compared to controls, participating in POS trafficking. And I put here, and therefore presumably cargo degradation. We haven't tested that, but that is something that I would, you would go on to assume perhaps, and that's certainly more work to be done. And this suggests a new mechanism through which A-beta can induce chronic RPE damage. So I'll leave it there, and I'd like to acknowledge these individuals. This work was mainly Savannah's. This is her PhD. She's doing a postdoc with me now. Some of the trafficking work and the characterization is done by Ellie, and most of the funding for this came from the NC3R. Thank you very much.
Questions in the front, please, Sam.
Very nice work. I was kind of curious, though, where do you think that the source of the amyloid beta is in the living retina and how it is being trafficked to the RPE? And I know that there's some publications that suggest that amyloid beta is accumulating in photoreceptor outer segments. I have some data that suggests that might not totally be the case, but I wanted to hear what your perspective on that was.
So there's been a number of studies, as you say, showing the accumulation of A-beta in photoreceptor outer segments in donor human tissues, also in the choroid. There's some evidence to show it accumulates in Bruch's membrane as well, and some work from one of the Alzheimer's mouse models have shown the presence of A beta in the RPE, but exactly where they haven't quite shown. And all of that can be reproduced in mouse models to varying extents. So that is what's known. And it's been thought for a long time that one of the major generators of A beta in the eye is the RPE. And our work, as well as others, have shown that the RPE expresses all the major isoforms of APP. And we see here actually that they produce ample amounts of A beta. Some others have taken RPE from young mice versus old mice, and they've looked at the amount of A beta secreted by RPE cells, although not a directional study, they just looked at what was in the culture. What was interesting is that RPE from older mice produced more levels of A beta compared to younger mice. So there's a whole raft of literature showing multiple A beta deposits in the aging retina.
The gentleman at the back.
That's fascinating talk, so thank you. Do you have any information on whether acid lipase activity is affected when you feed the amyloid?
Not to my knowledge. It's not something we've looked at, but I'm sure that there's literature there where they may have looked at this.
Because that would just suggest it. So your thinking is that it's just a general defect in acid hydrolases, basically, activity in these sort of congested lysosomes.
So the lysosomal response to this has been surprising. It's not something we've sought after. I know certainly in neurons it's been shown, and we've shown this as well, using acid phosphatase. This is some work that we published with Professor Lewis Serpel in Sussex. But it was difficult to get a traction on the dynamics behind lysosomal responses. There's very little known. It's one of those known unknowns, if I could use that word, that people know that various enzymes are trafficked into lysosomes. But then how do these lysosomes respond to cargoes? So the fact that there was an upregulation in cathepsin B was not surprising because a new cargo has arrived. But then what happens to it? Does it actually do the job? And this is that new information that really this study pulled out. And there's very little literature on that, but not specifically that.
And any effect on the acidification of the lysosomes? I think that could be measured as well.
Yeah, so that's something, one of the things that Savannah is doing. So one of the things that certainly come from the Alzheimer's world is that the acidification of lysosomes is driven by this VATPase, which also is the same mechanism that is underpinning synaptic vesicle recycling in the protonation of this, so that new neurotransmitter can be put in after release. And the idea is that this mechanism would be impaired, and certainly Ralph Nixon has suggested that changes in acidification and impairment in that creates the exact microenvironment within lysosomes for A beta to form these high molecular weight oligomeric forms that the lysosome cannot get rid of, and thereby creating a reservoir of toxicity. And this has been borne out by our studies as well as so many other studies, and it's difficult to tease these things out. They're fiendishly difficult experiments, but certainly, yes, that's a very interesting question, yeah.
Thank you.
And we may just take one more, just Rosie at the back.
I have a question about whether you saw any inflammatory cell infiltrate associated with the chorioretinal pathology in the mice. And the reason I ask that is because in humans, when you see this sort of localized chorioretinal inflammation, it's a uveitis. And uveitis is, of course, the more autoimmune type pathology, as opposed to sort of inflammation leading to neurodegeneration. So I just wondered what your thoughts were about that.
So Glenn Jeffries, your man, when you go back home, so he has shown that in the studies he's done that retinal microglia come up and mop up A beta in the retina. And you can certainly see them within these macrophages and these retinal microglia. So they certainly seem to do this, which is something they also perhaps do in the brain. whether there's an upregulation, I don't recall any literature. I think there are various mouse models that people try to quantify this, and it's not always easy, because this shift between activated and quiescent microglia is something that people are finding a bit difficult, because the idea is that suddenly they would have these ramifications and become active, and that's been shown actually that's not always the case. So I think that there's some difficulty in trying to see how it activates. But certainly, Glenn Jeffrey's shown that they certainly gobble them up.
Thank you very much.
Thank you for your
Thank you very much.
presentation.